Topic 7.1 is an outline of DNA structure. When covering this topic, it's important to emphasize that DNA is a double-stranded molecule and that its basic subunit is a nucleotide. Now, the students will be expected to be able to draw and label a nucleotide as well as a DNA molecule consisting of at least 2 pairs of nucleotides. Okay. So, for a nucleotide structure, students can draw something like this. Where the circle would represent the phosphate group. Is this visible? Phosphate group. The pentagon would be representing the pentose sugar, which in the case of DNA would be the sugar deoxyribose. And finally, the rectangle would represent the base, the nitrogenous base. Students also will need to be able to identify the direction of the DNA molecule. For this purpose, it's important to emphasize that in the pentose sugar, the carbon atom labeled carbon 1 is over here attached to the nitrogenous base. And an easy way for students to remember is that the phosphate group represents the 5th carbon atom, and this point over here represents the 3rd carbon atom. I'll come back to this point when I draw a DNA molecule in full later. With respect to the nitrogenous bases, students should be able to categorize the nitrogenous bases into 2 main groups: the purines and the pyrimidines. The purines are double-ringed nitrogenous bases, and there are 2 examples that they should be familiar with. They should be familiar with adenine and guanine. Pyrimidines are smaller, single-ringed nitrogenous bases. Once again, there are 2 examples that they need to be familiar with, which are thymine and cytosine. At this point, I would usually interject and tell the students that in the case of RNA, they will have another pyrimidine called uracil in place of thymine, and in RNA, once again, they will have ribose instead of deoxyribose. But this is to be covered again later under the transcription section. I'm going to just erase the board for a minute. Students will also be expected to be able to draw a DNA molecule consisting of at least 2 pairs of nucleotides. This is something like what a 2 nucleotide pair DNA molecule would look like. Now on this diagram, students need to be able to label direction. As I mentioned earlier, the phosphate group is always found on the 5' end of the DNA molecule. So put a 5' there, while the end which does not have a protruding phosphate group will always be the 3' end. Same for this side of the molecule, 3' and 5' ends. I would also emphasize to the students that they should note that the two strands that are found here are running in opposite directions from each other. They should also know that these bonds shown in the dark blue lines represent covalent bonds. But the two opposing strands are held together by hydrogen bonds, which I will represent in red. The hydrogen bonds are formed on the basis of complementary base pairing with bases, nitrogenous base adenine always pairing up with the nitrogenous base, sorry, I made a mistake there. The nitrogenous base guanine always pairing up with the nitrogenous base cytosine by forming 3 hydrogen bonds, while the nitrogenous base adenine pairs up with the nitrogenous base thymine by forming 2 hydrogen bonds. This complementary base pairing holds the two strands of the DNA molecule together. Once you've covered the basis of DNA structure, I would go on to talk about how the DNA molecule is stabilized by being wound around proteins known as histone proteins. Histone proteins, and how groups of 8 histone proteins are known as nucleosomes. These nucleosomes are important for the condensation of the DNA molecule to form a chromosome itself. Furthermore, I will elaborate to the students that the overall DNA molecule has a large portion of non-coding segments as well as coding segments, which will be important for the section on transcription later on. That would be it for 7.1.